The Array Studio Analysis tab includes a survival trait association function, also known as a Cox Model, which can identify variables that correlate with the length of time before an event occurs. In other words, we can find gene expression that correlates with patient survival. Several LAND datasets, including TCGA LAND and clinical outcome, include both expression data and survival data. LAND data can quickly be retrieved to a local project and used in a Cox analysis. Results from Cox analysis can then be confirmed in the LAND survival view. In this example, RNA-seq expression from TCGA will be retrieved for low-grade glioma patients. First, create a new project in the Analysis tab. Click New, New Local Project, or New Server Project. Let's call it Cox Analysis Gliomas. Now, in the LAND tab, let's apply some filters to TCGA to make sure we look exactly at the samples we want. We only want LGG, or low-grade glioma, primary tumor samples with RNA-seq data and survival data. First, filter for LGG under Tumor Type. Now filter for primary tumor samples because we only want to consider gene expression in tumors. Filter for data availability RNA-seq transcript. Finally, filter for survival status. We don't want NA or missing samples. Now, to ensure that our downloaded metadata include tumor type, survival status, and survival days, we will create a new metadata query for these three columns. Hold down Control to select multiple items. Now create a new sample set from a custom query. Select the query items and choose OK. Let's call this LGG Survival. Now let's download the data to local analysis. Click Download, Download sample data to local analysis, download selected samples across all genes. Choose your project, then select RNA-seq transcript unless you wanted to look at microarray data. Select gene level data and load your sample set. Now click Download. Unless you are requesting a huge number of samples, this should not take too long to retrieve the data. When the LAND data have been added to your solution, switch to the Analysis tab, where you should find an omic data object. This is an expression table, but with additional metadata attached for each sample and each gene. Downloaded expression matrix data, omic data, from array lens are usually stored in a memory-efficient data type, but this isn't compatible with many analysis functions. Right-click the object and choose Convert to Microarray Data. Although these are RNA-seq expression data, the expression matrix can be treated the same way as array expression data. If you wish, you can right-click and rename as LGG Survival Converted. If you open the design table for your omic data, you should see columns with survival status and survival days. The Cox Model function is found under Microarray, Inference, Cox Model. Make sure the right data are selected. Specify an output name. Then specify the model. In your metadata, choose the time and status. If you want, you can add additional terms to your model. You can output p-values for covariates and goodness of fit, and you can also report hazard ratios. When you're ready, click Submit. When the analysis is complete, you'll find the output under Table, Inference. The main table will have a list of genes with p-values. You can sort on this list to find genes that correlate with survival. Let's check some of these genes in the Land tab. For example, we can search for TGIF1 in TCGA land. Because we pre-filtered for LGG primary tumors and didn't release those filters have been inherited. Switch to the RNA-seq quantification RNA-seq survival view. Samples are automatically grouped into quartiles based on visible data. Quarter 1 is plotted as down, quarters 2 and 3 as no change, and quarter 4 as up. It is clear that high expression of TGIF1 in LGG samples correlates with poor survival. But if we clear the filter to look at all tumors, there's not a very strong correlation. We can also cross-check in clinical outcome land. Search again for TGIF1. Filter for disease state brain glioma LGG. And notice that low TGIF1 expression correlates with better survival.